Welcome to this webinar on teaching multivariate methods. This is the first one of a two-part series. My name is Kevin Pockner. I'm an academic ambassador on the JMP Academic team. A few things before we get started. If you need any help learning JMP, please, by all means, contact us. We're happy to do one-on-one support, group workshops, hands-on workshops, any kind of tutorials that you think will be beneficial for you and your students to learn JMP. We always love spreading awareness of JMP on campus, so if you have colleagues that you think would be interested in seeing what JMP is, please contact us and we'd love the opportunity. We, of course, have our webinar library. It has a large collection of webinars on a variety of topics. Encourage you to take a look at some of these other ones that may be relevant to your work and your teaching and research. We also have a collection of teaching and learning resources, including case studies, sample data, video tutorials, one-page instructional guides, e-learning courses, a lot of stuff to make using JMP in the classroom easy for you and also easy for your students to learn. So please take a visit to jmp.com/teach to see some of those resources. The agenda for this webinar. We're going to start off by showing a variety of tools to just deal with exploring multivariate data. We're going to look at correlation tables, scatter plot matrices, parallel coordinate plots, 3D scatter plots, heat maps. I'm also going to cover a statistical technique called clustering, where you group together similar or like observations. These tools are very useful for any kind of data set where you have a large number of variables and you want to understand and look for patterns and features. We're going to cover a technique called multivariate analysis of variance, or MANOVA, and that's where you make comparisons across many variables simultaneously. So it's the multivariate analog of ANOVA. We're also going to look at an example of repeated measures, and that's where multiple measurements are made on, say, the same subject over time. And we want to look to see if there are significant changes over time and also between some kind of treatment. And the last technique we'll look at is something called partial least squares. And this technique is designed to, when you want to build a prediction model, but you have a very large number of variables that are correlated with each other. And in most cases, there are many more variables than there are observations, and ordinary least squares doesn't really work. So in this first data set we're going to look at, there are 374 specimens of rice plants. And there are two categorical variables, country of origin and subpopulation. You can see that subpopulation has 6 categories. And the remainder of the variables are traits that are measured on each of these specimens. And all of these traits are numeric continuous. And at the very top of each column, you'll see this little column header. That's where you'll see a histogram and the min and the max shown when you have a numeric continuous variable, as we do here. This is a quick way just to get a sense of the data and the range that it's in and what the distribution looks like. Now, these traits are grouped into different categories: flowering time, morphology, traits about yield, seed morphology, stress tolerance, and quality. So I've just organized them over here. So if you haven't done this grouping of columns before, if you have a list of all the variables, all the columns, you can grab a set of them, and if you right-click, you have the ability to put them in groups. Now, I already have them in groups, so the selection here is to ungroup them. But that allows you just to organize your columns a little bit. So the first output that I'm going to create is just a table that's going to show the count of the number of specimens in each of these categories: country of origin and subpopulation. So I'm just going to create this table right here. I'm going to move it aside. To create a table, under the Analyze menu, Tabulate. And in this tool, you're interactively building this table. So if I grab country of origin and I drop it here on the row zone, it's going to start off by just showing me the counts, the number of rows in each of those countries of origin category. But I also want to bring in subpopulation, so I can pick that up and I could drop it right there. And then now I have the count in each of those combinations. And you can see there are many that we have 0. There's not a rice specimen of that country of origin and subpopulation. So I'm simply doing this just to get familiar with the data. Now let's start looking at these numeric continuous variables. So I'm going to launch the Multivariate platform. And this is where you put your numeric variables, put them all there. So you have the whole list. And I hit OK. And JMP's going to start off by showing a correlation table. Now, when you don't have many variables, JMP will also create a scatter plot matrix. But when you have a large number of variables like we do here, you have to ask for it. Now, this can be very cumbersome because with 30 variables, there is actually 435 pairwise correlations to look at. That's very difficult to do when looking at a numeric table. So JMP has an alternative way to look at this. And under the red triangle, I'm going to choose color maps, color map of correlations. Let me make this a little bit bigger so you can see it. And now we have a heat map of the correlations. And we can float our cursor over any of these cells and we can get the correlation value for them. And so here we see the more blue the cell is, the stronger the negative correlation, and the more red it is, the stronger the positive correlation. Now I'm going to make the scatter plot matrix, even though we have so many here, just so you can see. So again, there are 435 possible scatter plots you can make of these 30 variables. This gets a little difficult to look at when you have so many. So what I like to do instead is I'll create a scatter plot using the graph builder. And so what I'm going to do is I'm just going to select that first variable, put it on the y-axis, select the second variable, put it on the x-axis. I'll make this a little bit larger. Now I'm just looking at one of those possible 435 scatter plots. Well, what I'm going to do is I'm going to launch JMP's column switcher, and I'm going to pick each variable. So I'll start off with the variable on the x-axis. And now I've set up this column switcher over here. And then I'm going to do it again. And this time I'm going to choose the variable on the y-axis. Again, swap out with all of these variables. And now what I can do is look at any possible scatter plot by just selecting what I want on the y-axis and what I want on the x-axis. So you may find this more convenient than creating a scatter plot matrix with so many scatter plots, especially when they're so small, because now I have a large view and I can just focus in on the variables that I want to look at. Now, something else that's very convenient to do when you have categories like subpopulation is to create a different color or different symbol for each. So when we look at a graph, it will identify, in this case, those subpopulations. And you do that under the rows menu. You select color or mark by column. And I'm going to select subpopulation. Now JMP will pick a default, but I can right click on there and choose any color I want, but I'll stick with the default. I hit OK. And now you'll see over here in the data table, these row states are telling us the color of the symbol it will use. So if I go back to either the multivariate platform in the scatter plot matrix. Make this larger so you can see. It's using all the colors. But in this case, you may find it easier to work with the large scatter plot we created in the graph builder and then just choose which pair we want to look at. Now, if you're new to JMP, when you want to save output to the file, like in this case, this graph, you select the red triangle, save script to data table, you give it a name. I'll call it scatter plot, all vars via column switcher. It's a notation I like to use. Click OK. I'm going to close it. I'll close this one as well. And you'll see I just added that script right here. And whenever I want to see it again, I just click on it. Now, I already have it saved up here, so I'm going to delete that. Now, because these traits actually land in different categories, it might not be a bad idea to do the multivariate analysis for those groups. So if I select just the flowering time variables, and there's 5 of them, click OK. Now I'm going to get the scatter plot matrix and the correlation table, but in this case, there's only 10 possible scatter plots and 10 possible correlations to look at. You'll notice we have the subpopulations in the different colors. Something I often do when I have different categories, like subpopulation, is I'll actually bring up this data filter. I'm going to select subpopulation. And now over here on the left, I have this data filter that allows me to filter which data I want to use for this graph. I'm just going to make the graph just a little bit larger. So over here on the left, I can select, well, which one of these do I want to look at, or which collection of these do I want to look at for the purpose of comparison. And so you'll often find that a handy way to explore data. Another way I like to look at it is, and let me share the screen with the graph and the data table. Now at the data table, I can select populations, and when I do that, you'll see on the graph on the right, it grays out or fades back the other data points. So this is similar to the data filter, except I'm still able to see the other subpopulations there as well. And again, you can look at one or many of them. I already have this saved to the data table, the script right here, multivariate _ flowering time. But if it wasn't there, I'd go into the red triangle, save script to data table, give it a name, click OK, and that would be added. Now, under the red triangle is, of course, our contextual menu of options that you might want to do, in this case, to this scatter plot matrix. Here's the top red triangle for the multivariate platform. I'm going to add something called a parallel coordinate plot. I like this plot a lot. Let me just resize it. What this plot does is it shows all of the variables, but on the same scale. And so what you'll notice is the lowest value for each is all shown as the same. And here is the highest value for each. And now the data is spread according to that minimum and maximum. So it takes data that could be on very different scales and brings it on the same scale for the purpose of comparison. And I can select the data here. I can select the data up here, and you'll see that get highlighted. I'm going to make this a little bit smaller just because I want to get the data filter into view. And so that same approach is where I can look at individual groups or make comparisons between groups. You may also find it useful to look at the data in 3 dimensions. So I'm going to create a 3D scatter plot of the flowering time variables. It starts off with choosing 3. Spin this around. And then over here is where I can choose which variables do I want to plot, because I have other variables that I can look at. So you may find this a useful graph for looking for features and patterns in the data. JMP also has a tool inside the Multivariate platform that's very useful for identifying outliers or unusual values. Just from this scatter plot matrix, we might have noticed this value right here. That's row 81. That's a large value of flowering time in Arkansas compared to the rest of the specimens. Make sure I have all the subpopulations shown, and still that value stands out as larger than the rest. Well, what JMP can do is run an outlier analysis, and I'm going to choose this technique called the Mahalanobis distance. Basically what this is, and I'm going to close some of these other graphs just so this is the only one on the screen. Make it a little larger. Basically what this graph is, it's a measure each data point is from the centroid. So we have 5 different variables. So you can imagine all of these data points are essentially floating in 5-dimensional space. I know we can't actually see that. Here was the 3D scatter plot, so we can obviously see in 3 dimensions. So I'll use this to illustrate. There's a center. We call it a centroid. It's the multivariate mean of that cloud of points. We can measure the distance each data point is from that centroid. It's basically just a vector. Well, we can see that in 3 dimensions. We can't really see that in 5 dimensions, but it still exists. So this graph is plotting that distance on the y-axis. So we're able to see the points that are far from the centroid in these 5 dimensions. There's that observation right there. Let me open up the scatter plot matrix. That was that point. But what's interesting is there's another one right here that's also unusual in 5 dimensions. And you can see it on the edge on some of these variables. It's a little bit on the extreme. If I go to the 3D scatter plot, you can see that. Now, I know it might not seem as unusual as this orange data point, but remember, these distances are in 5 dimensions and we can't really see it. So within 5 dimensions, both of these points are almost equally unusual, far away from that cloud of points. So you may find this a useful tool just to get familiar with your data, look for some extreme values, identify some of those unusual observations. Now, I went forward and did that analysis for all of these groups. Here are the 3 morphology variables. Here's a set of yield component variables. The seed morphology. Stress tolerance, there's only 2 there. The 3 quality variables. So I often start off many analyses by going into this multivariate platform. It's just a great way to get familiar with the data, see if you can identify some patterns and features, unusual values, and in this case, make comparisons. And here we are looking at subpopulations, how they might be different. So this is a great tool. You remember at the beginning, we created this table that just showed the number of specimens in each combination of the country of origin and subpopulation. So similar to that tabulate, here is a heat map. So I have all of the countries of origin on the y-axis, the 6 subpopulations on the x-axis. And each cell is showing me the value of the specimens in those groups. For some, there's only one specimen. So for example, this observation here, row 292, the value is 106.6 for this flowering time at Arkansas. Now others will have more than one. So here's one here that there are 3 specimens in this data table in those categories, row 64, row 255, and row 256, and the average of those 3 values is 95.8. So I've created this heat map. Similar to what I showed in the multivariate platform, I created a local data filter for country of origin and subpopulation, and what that allows me to do is to look at smaller sets. So for example, maybe I only want to look at this group and these subpopulation categories, and then the heat map is made small to just represent those ones I've selected. So you'll find this local data filter very handy. It lets you look at data where you've got a lot of variables or categories, and you can work from one graph, one view, and then you just select the categories that you want to focus on and see. Now to unselect, you go into the red triangle, clear selection, and then now I'm back. Now over on the left, I set up the column switcher where I have all of the trait variables. So now I can just click through and see a heat map of any of these variables. And again, can also filter the data any way that I wish. You'll find this approach to JMP, this interactive approach, it allows you to just work with one graph, one view, and filter the data any way you want, switch the variables plotting any way you want, and it's so much more convenient than trying to create every possible combination you want to look at. I think JMP is really great at exploring multivariate data because of these 2 tools, the column switcher and the data filter. And the last analysis I'm going to run with these data is clustering. And before I do that, just want to point out that we have a recorded webinar entirely on clustering. So I encourage you to take a look at this. I'm going to be able to just cover it for about 5 minutes or so, but we have an entire webinar on that topic that I encourage you to check out. Now, before I run the clustering, and you probably noticed this when I was moving through the data table, you'll notice that there is many missing values in this data table. I'm pointing this out because the cluster analysis technique will not include a row if there's a missing data value. There's going to be many rows that won't be a part of this analysis, wherever there's a missing data value. Launch the hierarchical cluster platform. I'm going to put all the variables, click OK. And JMP starts off with a dendrogram. A dendrogram is a visual representation of the process of grouping together similar or like observations. And the way it starts, and I'm going to open up this clustering history here, is JMP is going to scan through all of the, in this case, the specimens where there's no missing values, and it finds which 2 observations are the closest. And it turns out it is row 212 and row 227. That is the first 2 rows to join. And then it keeps on scanning. And then it finds the next 2 closest observations and it joins those. Now what happens is either an observation is going to get clustered with another observation, or an observation is going to get clustered with an already formed cluster made up of many observations. Or it takes a group of observations in a cluster and joins it with another group of observations in a different cluster. And so the dendrogram is representing that. Now I'm just going to zoom in because there's many observations just to get a closer view. And so just looking at this group, here is 212 and 227. Those are the first two to join. Now you'll notice not long after, row 233 joined that cluster. And then 226 joins that cluster, then 216 joins that cluster. Then that cluster gets joined with this cluster over here to form this one. And then that group gets joined with this group over here, and that creates this cluster. And so this process goes on until eventually everything is in one cluster. And the distance that these lines make up is representing how far those points are away. And let me get back to seeing the whole dendrogram. Now, right now, if you remember, we set up the colors of the rows are based upon the subpopulation. I'm going to change that, and instead, I'm going to color the rows by the cluster. So when I choose that, it's now changing those row states. Now, you can interact with the tool to decide how many clusters you want to look at. So here I am dragging this around. At what point do I think these observations have been grouped into a reasonable set of clusters? Now, there's no specific answer, and since this is an unsupervised learning technique, we have no way to evaluate if we're clustering well because we don't have an outcome variable we're predicting. So we typically look to see when is the distance between clusters start to get large. And the common approach is to choose a number of clusters where you see that plot make a large inflection. So a combination of that, as well as looking at these distances in the dendrogram, you start to get a sense. Sometimes it's driven by a predetermined solution. You want to group the data into 5 clusters. So let's just say that's what we want to choose, 5 clusters. I can describe those clusters by selecting Cluster Summary. And now each one of these lines is representing the mean for each one of those variables for all the observations that are in each of those clusters. And I could look at those mean values by opening up that table. I could even look at the standard deviation, so that's showing me the variability in each of those variables within each of those clusters. And if I wanted to see the individual observations in each of those clusters in this parallel coordinate plot, I can ask for that. And so now I can look at all the observations that make up each of these clusters and the values across those variables. So this parallel plot over here on the left is showing me the mean, and this parallel plot over here on the right showing me all of the observations in each of those clusters. Now, if I wanted to incorporate the subpopulation and see how the subpopulation compares to these clusters, I can ask for that. Choose More Color Map Columns. I pick the column, in this case subpopulation, and then now over here on the left, I'm seeing the subpopulation using colors to identify them, and then I can see how much does the clustering actually correspond or match to the subpopulation. And I can see some of that. There are places where you can see that the specimens that come from a particular subpopulation group together. But of course, there's others that aren't. And those might be worth exploring because they're not similar to the other observations in that subpopulation. I can also select to cluster the columns. So when I select two-way clustering, this dendrogram gets added with a clustering of the variables. And so that's grouping together variables that are correlated. So if you look, variables that cluster together early are variables that have a high correlation. And so I'm seeing that clustering. And then a heat map is made. So each one of these cells in the heat map is a cell in the data table. And I can float the cursor over and see the value. And they're just organized now. So on the y-axis, I'm clustering the observations, and that's creating the rows. And then on the x-axis, I'm clustering the columns, and that's creating the columns in this heat map. So you may find clustering a useful tool to understand the data a little bit more and see observations that are like other observations, or maybe observations that are most unlike other observations, the unusual values and outliers. Okay, so I'm going to close this output and this data table up, and we'll move to the next example. Now we're going to illustrate JMP's MANOVA platform, and we're going to use two examples. So for one example, we're going to make comparison across groups when taking into account many response variables simultaneously, and the other example is going to use the MANOVA platform to do repeated measures. So in this example, measurements were made on 6 different chemical properties in water at 3 different locations. So the objective of the analysis is to find out, are there differences in these chemical properties between the 3 locations? And so an initial analysis one might do is to do individual one-way ANOVAs. I'm going to do this 2 different ways. One is through the graph builder. So here I'm going to put the first chemical property as the Y, location as the X. Starts off with just a dot plot, but I can choose to add the mean and confidence interval for the mean. And over here I have the controls to actually add the F-test. And here I will see the F statistic along with the p-value. Now I'm going to set up a column switcher so I can bring in the other variables. Now keep an eye on that F statistic and p-value. As I move through the different responses, you will see that none of them result in a statistically significant p-value. So one might be led to believe that there are no differences in the means of each of these chemical properties between the 3 locations. Now, if I wanted more formal output for one-way ANOVA, I would use our FitYBX, our bivariate platform. What I'm going to do is I'm going to do them all together. Location, click OK. Now JMP's default is to stack them, but what I'm going to do is put them side by side by just choosing Arrange in Rows. There are 6 of them. Click OK. And then now I have the results for each going across horizontally. Now under the red triangle is where I could choose means ANOVA. If I select that, I will get the results, in this case for chemical property 1, added to that column. And I want to do the same for the other 5 chemical properties. Now I have the ability in JMP to do it all together by just holding down the command key on an Apple computer. And now when I select an option under this menu, it's going to do that analysis for all the variables. Now these are the same results, same F statistic, same p-value. I just want to show you the bivariate platform for those of you who aren't familiar with it. This is where you would do a more formal one-factor analysis of variance. Here I'll sorts of options that I might do, like multiple comparisons, diagnostic plots, etc. If I run the multivariate platform, which is where I'm going to produce the correlation table and the scatter plot matrix, I'm doing this to try to motivate what's going on in MANOVA. Now here I'm just looking at two variables at a time. It's basically comparing the multivariate mean. So you can think with each group of these points, there is a mean of each of those two variables. It's called the centroid. It's the multivariate mean. MANOVA is making a comparison between those. And it's not just taking the variation into account, the within group variation, like one factor ANOVA does. It's actually taking in the correlation into account as well. I'm going to make the scatter plot matrix in a different way because it's going to give me an option that I want to add to the graph. So under the graph menu, choose scatter plot matrix, put all the variables. I'm going to choose to show both the upper triangle and lower triangle. And what I'm going to do is I'm going to add a density ellipse. I'm going to choose the outline as well as the shaded. Now this is doing it for all groups together, not separating the groups. I can separate the groups and get ellipses for each by selecting group by, and I want to group by the column location. I can also change the coverage area. What I'm going to do is I'm just going to choose 50%. I want to make these ellipses a little bit smaller to make it easier to see what we're comparing. Click OK. And now I'm getting a two-dimensional ellipse for each of the groups. Again, what MANOVA is doing is it's looking at the mean, the centroid within each of those ellipses, and it's comparing those, taking into account the variation and correlation. And you can see there's some correlation among these variables, some more than others. Look at the correlation between C2 and C3. So just looking at it in two dimensions, you might start to get a sense that there are some differences. Look at these two variables here. Does the centroid of the blue ellipse to the red ellipse to the green ellipse, are those statistically different? So that's kind of what's going on. Again, we're going to do MANOVA in 6 dimensions, so we can't really see that, but I'm just trying to motivate what's happening. And I'll do one more thing to motivate it, and that is to create a 3D scatter plot. Choose all 6 variables. I'm going to make this a little larger. I'm also going to make the points a little bit larger by just right-clicking, choosing settings, and adjusting the marker size. And here I've got a 3D scatter plot, and I can click through, so it's going to go through every possible 3 dimensions. And I'm going to add density ellipsoids. And similarly, I'm going to choose to show ones for each group as opposed to an overall ellipsoid. Click OK. Now we're getting the ability to compare in 3 dimensions. So what MANOVA is doing is the multivariate mean in 3 dimensions different between these 3 groups when taking into account not only the variation but the correlation. And here's another 3 variables, property 4, 5, and 6, just so you can get that idea. Now again, we're going to be doing this in 6 dimensions. We just can't see 6 dimensions, so I'm using these graphs as a way to motivate what's happening in MANOVA, and they might be useful when you're teaching it in class to help the students understand what's going on in MANOVA as well. That's why I decided to show these. So to run a multivariate analysis of variance, we launch our fit model platform. Put all the response variables as the Y. For the personality, you want to choose MANOVA. Now we only have one factor at 3 levels, location, so we'll add that as the model terms. But we could have multiple factors, in which case we'd be putting them all in and possibly interactions as well. But here we just have one factor. So I click run. This initial set of output is actually one-factor analysis of variances. So I've got individual models for each response. So this is a one-factor analysis of variance model. Or maybe more convenient is to look at the estimates of the means here, and I have some graphs corresponding to that. Again, this isn't MANOVA yet. This is just some initial output JMP is starting with. So I'm going to close these. And what I need to do is choose a response. Now, we have different comparisons we can make. If you float your cursor over each one of these, there'll be a little bit of a description. If we're doing a basic MANOVA, we want to choose the identity. You click run, and JMP is going to start off with doing some statistical test. We've got a whole model test, which is actually going to be the same as the test for the factor location, just because we have one factor. But if we had multiple factors, those would be different. And then, of course, there's the intercept test, which in many cases is something we're not interested in. So I'm going to close some of these up. Now, when you get output in JMP like this, you might be unfamiliar with what this output is, as well as pointing this out to your students. Well, a convenient way to learn some more about the output is to click on the question mark at the top and float your cursor over, in this case, this table. And then when you click on it, you'll be sent to our help documentation, in this case, on MANOVA. There'll often be descriptions of mathematical details. There's often an example of a data set interpreting output. I also came across this post in our community about the different MANOVA test statistics. It's a really nice coverage and discussion about what these test statistics are. And it's talking about the mathematics, but in a really accessible and fun way. We also have this book we produced, JMP for Basic Univariate and Multivariate Statistics. And this book covers MANOVA, both in the comparison technique as well as repeated measures. Regardless of what statistical tests we use, you'll notice that they're all significant. In other words, we now have the evidence, the statistically significant evidence, indicating there's differences in these chemical properties between the three groups. It's just that it's done in a multivariate sense. So we can't say that difference is in one or some of the chemical properties, but it's in them collectively. Now we're going to explore this a little bit more to see if we can get some insight to describe what those differences are. One thing we can do is start doing individual comparisons. So I'm going to make comparisons between A and B. A and C. And B and C. And you'll see here that there's a statistically significant result for the A-B comparison, but not for the AC or BC comparison. Now, what's going on mathematically in the background is JMP is creating new variables, composite variables, linear combinations of these 6 chemical property variables, and they're called canonical variables. And so I'm going to start off by saving the scores for those. They get added to the data table. And if you click on the columns and look at the formula, you will see what that linear combination is. It's similar to the idea of principal components, except principal components creates composite variables that describe the most variation across the variables, where the canonical variables are creating linear combinations such that it's creating the biggest difference between the groups. You can get a view of those by asking for this centroid plot. Let me resize this report so I can make this a little bit larger. I can add the data values. So 2 new variables were created. They're independent of each other, hence why the density ellipses are actually circles. There's no correlation between the canonical variables, and they're created such that it creates the biggest difference between those 3 groups. And we can see that. And here is a graph of each canonical variable to make the comparison between the groups. And here again, you can see those differences. The challenge is interpreting them because these are new dimensions, a new representation of the data. Looking at the 2 canonical variables together, here's a scatter plot matrix with the density ellipses. Again, we're seeing that there are differences. We couldn't pick that up in a univariate sense, but with respect to these canonical variables, again, this is illustrating how it's picking up that difference. Again, I know it's quite a bit harder to describe what's happening when we're doing one-factor analysis of variance. It would have been easy if we can say, oh, chemical property 1 is different between the groups, or chemical property 3 or 5, and then which groups are different from others. This multi-dimensional sense makes it harder to translate that into a practical interpretation of the result. But it certainly gives us some insights. We can go down, drill down, look at more graphs and plots in multiple dimensions to see if we can describe that. But the key is that it's taking the correlation among all these variables into account, not just the variation as one-factor ANOVA does. Okay, I'm going to close this example. In this next example, we're going to use the MANOVA platform to do a repeated measures analysis. That's where we're going to make multiple measurements on the same subject over time. This study consists of 20 subjects, and 5 subjects were each randomly assigned one of 3 treatments, A, B, and C, and 5 were assigned to a control. And at 6 different time periods, their cholesterol level was measured. Now, these responses are going to be obviously correlated because they represent measurements on the same individual. So certainly if an individual starts off with a very high level of cholesterol, that may continue even though they're receiving a particular treatment that's reducing it. And we could calculate a correlation and look at a scatter plot matrix to see that. But I think what will be better is to create a graph that shows the change in cholesterol over time. So I'm going to use the graph builder. I'm going to drag the 6 variables and put them here on the X. Now, right now, it's just showing the individual points. So if I grab treatment and put it as the overlay, then that line is going through the mean of the 5 measurements at each time period in the 5 different subjects. But instead, what I'm going to do is I'm going to put the subject as the overlay and treatment as the color variable. Let me make this a little bit larger. And so now each one of those lines is a subject and their cholesterol level at those 6 different time periods. And I'm using the color to represent which treatment it was, A, B, C, or the control. Now, it's very obvious to see that the cholesterol level is not really changing for those in the control. It is changing for the treatments, but we want to go and find out where are those differences. Which of those drug treatments seem to be doing a better job at reducing cholesterol level? You can get a sense of that just by looking at the graphs. If you follow those points, for example, treatment A, we see there's a big drop going from time period 2 to 3. it seems to go relatively level from 3 to 4 and then drops again from 5 to 6. Something similar happens with treatment B, that's the red lines. And the green lines, that's treatment C. It looks like after a drop to time period 3, there's not much of a drop, if any at all, going to time periods 4, 5, and 6, unlike what's happening with A and B. So we're kind of already getting a sense that the differences might be between A and B and C, and that A and B might not be different. Now, clearly the control is different. I can obviously see that from the graph. So what I'm going to do is I'm going to remove the control from the analysis. I just want to make the comparisons between A, B, and C. It's clear that control is different than all three. So if I click on control, and I'm going to choose hide and exclude, now those 5 patients aren't going to be included in the analysis. You may notice that we have this column here, which is showing images of the subject. I'll make it a little bit bigger so you can see. So you can actually drag in images into the cells, into this data table. And what I did is I chose to make them the label. So now, I'm going to go back to the graph. If I float a cursor over a particular point, I can actually use the image of that subject as the labels. So to do a repeated measures, we launched the Fit Model platform. We put in all the responses. We choose MANOVA. Now we put in our factors. In this case, we only have one, which is treatment, but we could have more than one, in which case we might have main effects and interactions. But here we just have the one treatment, and we're looking at it at only 3 levels because I've excluded the control group. Click run. You're going to get initial set of output, and these are analyses on each. So we've got a NOVA model, a one-factor NOVA model for each time period. We can see the means, the estimated means for each group and time period, and we have some corresponding graphs. But I want to do a repeated measure analysis, so I'm going to close all these. And for choose response, I'm going to select repeated measures. JMP prompts you to give a name to represent what those repeated measures are. It doesn't have to be time. Time is something that's used most frequently. That's why it's the default word there, but it could be something else. I'm going to stick with the default name of time. Click OK. And some statistical results get added to this report. I will start off by looking at the between subjects. So here is where we're making comparisons between the subjects. And the model will consist of an overall test, an intercept, and a treatment. And since we only have one treatment, then the treatment is going to be the same as the overall test. And the intercept is not really a value, so I'm going to close up those just to look at the treatment. So right here, this is telling us there's a statistically significant difference somewhere between those 3 treatments, A, B, and C. To do those comparisons, I can choose contrast, and I'll select AB, AC, and BC. And now we can see that the difference is picked up between A and C and B and C. And we're not seeing a statistical difference between A and B. I'm going to bring up the graph again so you can see. We saw that in looking at the graph. So A is the blue lines, B is the red lines. That's where we're not picking up a statistical difference. And the difference is between A and C and B and C. The other analysis that gets done is comparing the change in the response within subject. So I'll open that up. I'll slide this over so I can bring up the graph at the same time. So clearly there's a change over time. We can see that. That's quite obvious. So the statistical tests are going to pick up change over time, and that's shown here. But what's of interest is this interaction term, time by treatment. So what's happening here is a test of, is the change happening over time the same between the treatments or different between treatments? That interaction effect. And we're getting a significant result. And I could choose to do contrast under here. And I will do A and B. A and C. And B and C. And so here we see the comparison of A and B, not different. So the change in the response over time is not different depending upon if it's treatment A or B. That change is similar. However, we're seeing, comparing A and C and B and C, that change over time is different. And again, we can see that in this graph. We watch how the cholesterol levels seem to drop similarly between time period 1 and 2, similar between time period 2 and 3, similar between 3 and 4, and it's really going from 4 to 5 to 6 is where we're seeing differences between those treatments. Treatments A and B have a drop going down to 5 and sort of level to 6, but treatment C doesn't seem to change going between 4, 5, and 6 time period. And because we are building a model, we can choose if we want to save the predicted values and the residuals. And I did that. I have the columns hidden, so I'm going to unhide them. And so here are the predicted values for each time period and the residuals for each time period. So I can do some model diagnostics if I want. Here I'll open up, I made a graph of the residuals for the treatments. We just have treatment A, B, and C. And then with the column switcher, I can switch between the 6 time periods and look at the residuals and maybe identify ones that stand out. We can see the variation between these time periods gets larger as we go further on. So repeated measures are very common in medical studies. Many studies are designed this way where the same subject is either measured over time or maybe gets different treatments. They might receive treatment A, and after some period of time, they might receive treatment B, and might receive treatment C. And there's a compounding effect going on between those treatments. So you'll see this type of statistical model in medical studies. It's not the only application they occur. Okay, so I'm going to close up this example. In this last example in the webinar, we're going to illustrate partial least squares. And this is where we're going to build a prediction model with a large number of highly correlated predictor variables and few observations. Before I show the example, I want to point out this book we have on partial least squares. I encourage you to take a look at this book if you think this is a topic, a statistical technique that would be very useful for you. So in this example, we have 6 specimens. Each specimen has a certain level of concentration of a component. These first 2 specimens, it's 0.085, these 2, 0.093 and 0.098. Now a spectrometer was used to produce this data. So we have these intensity values across 91 different wavelengths. The purpose of this analysis is to see if we can build a model, a prediction model that uses these intensity values across those 91 wavelengths to make a prediction of the concentration amount. Now, I'm going to switch to a data set that has that data stacked. And the reason why is this is going to be a little easier to create a graph I want to show first so you can get a sense of what this data looks like. Now here is a plot of the data, the intensity versus the wavelengths, and you'll see for each concentration level there are two specimens. I'm going to choose to represent this data with a smooth curve as opposed to the connect lines. So this is just giving you a sense of what the shape of this data looks like. In fact, I'm going to turn off the points just to make it a little clearer. So you see there are two lines for each concentration level, and those represent the two specimens. And the objective here is how can we use those curves, the information from those curves, to make a prediction of what the concentration amount is. Clearly, each concentration has a unique shape. Now, within a given concentration, the specimens aren't that much different. Those shapes of those curves are quite similar. So back to the data set that has each variable as a column, and this is how we need to have the data, not stacked. I use the stacked data to make that graph so you can see the shape of the data. But this is the format we need in order to build the prediction model. Just to help motivate why this problem needs a more sophisticated modeling technique, I made this graph where I'm plotting the concentration amount on the y-axis versus the intensity level on the x-axis for a given wavelength. And I have the columns which are over here. So for example, you can see that, oh, there's a little bit of a correlation there. And I can move through each of those wavelength values. And you can see there are correlations between those intensity values and the concentration amount. But they're very different. For some it's positive, for some it's negative, some it's strong, some it's weak. So it becomes challenging, well, which ones would I use to make a prediction? Using only one or a couple wouldn't be great because there's so much structure to that data that I would be overly simplifying it. If I try to use ordinary least squares regression, which I'll do so, I'm going to put the concentration amount is what I'm predicting. I will put all 91 variables here. If I try to run this, well, it's not going to work because I've got 91 predictive variables I'm trying to fit in here, but with only 6 observations. So there's nowhere near enough information. There's nowhere near enough data to fit that many terms in the model. And then just selecting one or two or a couple, that's just too challenging because you can't decide, well, which ones should I pick? The other aspect of this data that I want to point out is how highly correlated these variables are. You can clearly see that wavelengths that are close together are going to have similar values. Here's a heat map of the correlations of all these variables so you can see it this way. It's very clear from looking at either the correlation map or the curves that variables that are close together will tend to have similar values. So even if we had a case where there were not as many variables and we did have a lot of observations, ordinary least squares would still have a problem because of this high correlation. So partial least squares is found under the multivariate menu. Partial least squares. The response variable is the concentration amount. And all of those wavelengths are my predictor variables. And click OK. JMP has 2 model fitting methods. I'm going to leave the default. Click Go. Now before I start interpreting this, in JMP Pro, you can also access partial least squares from the Fit Model platform. So if I go in here, you'll see that partial least squares is one of the options under the personality menu. And JMP starts off with producing output for one factor. So creating one composite score, one variable that's a linear combination of all the candidate predictor variables. We can choose to look at more than one factor. So I'm going to choose 2, click go, and then results of fitting one factor or 2 factors are added to this report. Remember, to get assistance in understanding statistical details in this report, you can click on the question mark and float your cursor over different parts of this report, and you will get sent to our help documentation. But for our analysis, I want to focus on these 2 graphs right here. I'm going to make them a little larger. So this is showing a plot of these new composite variables, these scores, against the outcome variable. Now the y-axis isn't the actual outcome values, but a standardized version of it. But what I want you to notice is, look how strongly correlated that first factor is to the outcome. Very highly correlated. Not as much with the second one, but there is still some correlation there. It looks like this factor, this first factor, is really good at predicting the outcome. And we can quantitatively see here how much of the variation in the x's in the predictive variable this factor is explaining. And then here is how much of that variation is this factor explaining in the outcome. That's almost 100%. When we go to 2 factors, we explain a little bit more, not too much in the y. We explain a little bit more the x variation. So this is pointing us to believing we really only need one factor. One factor is going to be really good at predicting, in this case, the concentration. Now, to get a sense of what this one factor looks like, I'm going to close up some of these windows and show you the coefficients. So here's a graphical representation of the coefficients, the weights that are being put on each of these variables to create that new composite score that's a linear combination of them. Now, it's not that we really need to know what those coefficients are. That's not really the purpose of this analysis, to get some interpretation in those coefficients. What we're really trying to do is build a model that predicts really well. So it's okay we don't have interpretation here. We can ask for some more information, like here we can ask for a variable importance plot. And this is giving us a measure of the importance each of those variables has in that final composite score to make that prediction. But again, this isn't of primary interest here. It's not necessary that we're interpreting each of these predictive variables and how they end up making the prediction. What I'm going to do is I'm going to save the prediction formula into the data table. Now at the end, I have a new column. That's the prediction of the concentration based upon this composite score, this factor that's created. If you want to see what it looks like, one way is to go into the column and see the formula. So there are all those weights. As you can see, it's a linear combination of all of those variables that produces the prediction. Now, what might I do with this? Well, let's say I made measurements on 6 new specimens. But here I don't know what the concentration is. I just have the results from measurements from a spectrometer. Well, what I can do is I'm going to add 6 rows to this data table. And now I can grab these data and I'm going to paste them in. Go right over here, choose paste, and this last column, as you remember, there's a formula here. And so it's going to apply that formula to these data and produce these new predictions. So for these 6 new specimens, of which concentration I don't know, see, the data I used to build the model, to train the model, I knew the concentration. These I don't. But based upon this model I built through partial least squares, this first specimen is estimated to have a concentration of 0.0845. This one, 0.0988, 0.0885, 0.0922, 0.0982, 0.092. As you see, if the intent is just to make really good predictions, I can rely on a technique like this that's somewhat sophisticated mathematically, certainly way more than linear regression, but linear regression can't help me here. A technique like partial least squares is what can take this complicated problem, a very large number of highly correlated predictive variables with very few observations, and create a model that makes that prediction. There's also a more thorough presentation of the topic I found in the community. So if you go into the JMP user community and search by this title, you'll find a collection of tutorials with one of our systems engineers teaching the method of partial least squares. Well, I hope you found some value out of the webinar, learned some new statistical techniques, maybe some new capabilities in JMP. Don't hesitate to reach out to us if you would like some one-on-one support or presentation or workshop for you and your colleagues. And take a look at our teaching and learning resources. We really try to make it easy for you to use JMP in the classroom and for students to learn JMP. Well, thanks for watching and have yourself a good day.